Welcome back. This video is about DNA replication. Replication happens in that S phase of interphase in the cell cycle, where we take our chromosome from looking like this to looking like this, right? So all we want to do is duplicate all this DNA so that we have two of them. Don't be fooled by this single look. It is still a double helix with two strands, okay? So we're just gonna open these strands up and copy a new strand for each of them. So really there'd be 4 strands, 2 for this one and 2 for that chromosome. Let's draw some DNA. All right, so we have our double helix and we open it up in the middle at a place called the origin of replication. Just meaning where we're going to start. Our chromosome is really long and we're actually going to have several origins of replication. So lots of these bubbles and they'll just come together to kind of expedite the process. All right, we can draw some nucleotides on our DNA, or representation of such. Right, so those are like guanine, adenine, thymine, cytosine, etc. We also need to make sure that we put a 5' and a 3' in so we know which direction the DNA is traveling. Okay, so 5' to 3' just refers to that carbon number on the sugar. So remember we have a 5-carbon sugar in our nucleotides. So this is carbon 1, 2, 3, 4, and 5 is up here. And so attached to number 3 is a phosphate group, which will then attach to the next number 5 carbon. And it'll attach to the next 3, etc. So like this 5 will attach to a phosphate, which will attach to the 3rd carbon, etc., etc. So 5' to 3' means it's running in this direction. The strands are anti-parallel, meaning they run in opposite directions. So this one's 5' is over here, and its 3' is over there. Okay, so this strand's going that way, and this strand's going this way. All right, let's get some enzymes involved. Our first enzyme, we'll put them here, is called helicase. And the job of helicase is to unzip DNA. So it's the one opening it up. I'm only going to draw one on this side, but know that it's also happening on the other side. So it's opening it in both directions. Right? I just don't want it to get too crowded because as you're about to see, the whole thing's going to get really crowded. All right, let's get another color. Now, when you open your DNA, it wants to go back together. No barking. Sorry, that's my dog. These were hydrogen bonding to these, and we've ripped them apart, and they're trying to go back together. So we need to keep them separated with what's called single strand binding proteins. We normally just write SSBP. And they hold the DNA open. Okay, we'll put some up here too. We're gonna hold them open on this side as well. Right, same thing would be going on over there. And that's just so it doesn't close back on itself. Now, whenever you take 2 strings, such as DNA, and you twist them together, right, and you open the middle, down here, it's gonna get kinked up really tight because you open this, so that has to get tighter and tighter and tighter. And so way downstream, we have another enzyme whose name is topoisomerase. That's a fun word, topoisomerase. and its job is to unkink the DNA, right? So it's actually just gonna clip it, let the tension release on it by letting it spin, and then it'll glue it back together. And again, same thing happening on that end. So we already have 3 different enzymes doing work, and we haven't even done any replication yet. Now, when we put down new DNA, it always has to have a 3' end to attach to. And so if you don't have that 3' end, you can't put new nucleotides down. And since we're starting a brand new one, there's nothing to attach to, all right? Oops, I erased origin. What we do instead is put down an RNA primer. So I don't have enough nucleotides, but usually it's like 6 to 10 nucleotides long. And it's just somewhere that we can start because the RNA doesn't need a 3' end to attach to. It can just go anywhere. There is an enzyme who puts this primer down. I'm just gonna draw it like this. Its name is primase. Enzyme who puts down a primer. All right, so if our strand here is going this way, then that means our new strand that we're building is going to go this way. Okay, so we can attach to this end. And continue to go in that direction. Let's actually make that a new color of blue just so you can see that it's new DNA. I have all these lovely colors that a student gifted me, so let's put them to good use. Okay, so here's our new strand. We can't draw any over here because again, they don't have anything to attach to, right? So this is our new 5' headed to our 3'. This strand is going this way, so we are going to make our new one going the other way. So now we have 5' to 3'. All right, now because this strand can just continuously grow as we like open this DNA up, we're gonna keep growing that strand in further and further directions. It is called the leading strand. So the one that can just keep going is our leading strand. This is a leading strand and this is a leading strand. But what are we gonna do about the rest of them, right? If this DNA is opening in this direction, but we can only put DNA down in that direction, then we can't really add it that way. So what we have to do is put down little pieces to try to keep up. So we're gonna put little primers down. I'm just gonna put one line because I don't really have room to draw multiple, right? I'll put a primer here, let's put one here, okay? Just pretend it's a few nucleotides long. And so this DNA is opening in this direction, but we're trying to lay DNA down going that way. So we had to put a piece down, put a piece down, put a piece down, put a piece down to try to keep up, right? So we put our primer down, and now we can put more pieces of DNA down from the primer. Okay, so our enzyme, running out of room to write stuff. Our enzyme who's putting down these new pieces of DNA is called DNA polymerase 3. That's just who's putting down the new DNA nucleotides, okay? And we can put some on our other strand as well. Is this the right color? Hope so. Yeah. All right, we can attach there, attach there. And so these little pieces that we're putting down are called Okazaki fragments. Well, I hope that's not cut off on the video. Okazaki fragments. So that's just the little pieces that we're putting down on the opposite strand, whose name is the lagging strand. And that's because it's lagging behind because it can't make a continuous set of DNA. It can only make these little pieces. And so you'll notice that we still have RNA in there. And we can't have that, right? Our DNA is not like part RNA. So we need yet another enzyme. Enough enzymes already, huh? Another enzyme who's going to come around. I'm trying to find another color here. I don't have enough different colors. And remove this RNA primer and replace it. I'm going to go ahead and erase it out here. It's going to remove the primer and replace it with DNA. Okay. Its name that replaced it is, let's put it this way, DNA polymerase 1. And it replaces RNA with DNA. I don't know if you can see the very bottom down here. Hopefully you can. So it's going to come in and replace all these places where the primer is. But you'll notice that there's still a little gap because DNA polymerase 1 doesn't have the ability to connect these. So what color didn't we use? Brown, I guess. So there's another enzyme who's going to come in and connect them. Okay. His name is DNA ligase. And it is just the glue that glues these together, right? So it'll come in, the DNA polymerase 1 will come in and replace this. Ligase will glue them together. Replace this, glue them together. Replace, glue, replace, glue, replace, glue. Okay, so polymerase is replacing, ligase is gluing. And it will just continue all the way until the whole thing is copied. And then we'll have 2 strands. So our original 2 strands plus our 2 new strands to make it double-stranded. And of course, those will then become a helix and twist around each other, right? So each chromosome now has an old strand and a new strand. And that's it. That's DNA replication.